Have you ever stopped to think about the incredible hidden defense system working tirelessly inside you, fighting battles you never even notice?
It's pretty amazing. A constant silent war against countless invaders, viruses, bacteria.
And right at the heart of this whole complex system, leading the charge, are these unsung heroes, antibodies.
Yeah, exactly. The term antibody comes up a lot, doesn't it? Especially with vaccines or tests. But the sheer complexity and precision of these molecules, it's genuinely astonishing.
They're not just simple defenders, are they?
Not at all. They're like masterpieces of molecular design, built with this almost unbelievable ability to recognize and, neutralize specific threats.
And that's exactly what we're diving into today. This deep dive will pull back the curtain on the complex world of antibodies, taking us way beyond the basics. We've gone through a comprehensive set of sources detailing their intricate structure, their diverse functions across the body, and the incredibly precise, almost orchestral ways they're produced.
It really is like an orchestra.
Right. So our mission today is to give you a true shortcut to being well informed on this absolutely essential topic. We want you to understand what makes antibodies so effective, how they actually work their magic, and why they're so crucial for your health. So, ready to unravel this molecular mystery?
Absolutely. Let's do it. To really appreciate these molecular marvels, we need to start right at the beginning. What are antibodies, fundamentally?
Okay.
Think of them as specialized protein molecules. They're actually glycoproteins because they have these carbohydrate chains attached, and they're meticulously produced by a specific type of white blood cell, the B lymphocytes.
E cells, right.
Exactly. And they always spring into action in direct response to something the body sees as foreign or potentially harmful, something we call an antigen.
So an antigen is basically the bad guy trigger. And once an antibody is made, its mission is incredibly specific.
Incredibly specific. It's designed to recognize and bind only to the exact antigen that triggered its creation. It really is like a perfectly crafted key fitting only one unique lock.
That's a great analogy.
And it's this precise binding action that ultimately allows antibodies to protect us, the host, from all sorts of microbial pathogens, bacteria, viruses, even toxins just floating around.
Okay, so that precision is key. Now, you mentioned something important there, the terms antibody and immunoglobulin.
Yes. That's a crucial distinction, and one that often gets glossed over, even in the, scientific discussions sometimes. We tend to use antibody and immunoglobulin interchangeably.
Yeah, I think I do that.
Most people do. But immunoglobulin, or IG for short, is actually the broader, more encompassing term. All antibodies are immunoglobulins, structurally speaking.
But not all immunoglobulins are antibodies.
Exactly. Not every immunoglobulin functions as a protective antibody in the way we're discussing today.
Yeah.
For instance, there are certain abnormal proteins found in some diseases. They're classified as immunoglobulins, but they don't actively fight off infections.
Okay.
So think of immunoglobulin as the big family name and antibody as the specific member of that family that's really geared up for defense.
That makes perfect sense. Like saying all terriers are dogs, but not all dogs are terriers.
Precisely. Good analogy.
And, to really grasp how we even got to this understanding, it's fascinating to trace the history, isn't it? Imagine trying to figure out something invisible that held the key to health and disease.
For centuries, it was a total mystery. Then, back in 1890, two pioneers, Emil von Behring and Shibasaburo Kitasato, they really cracked open the door with a revolutionary discovery.
What did they find?
Well, their work was foundational because it shifted the whole scientific thinking at the time. Many believed immunity was purely cellular, just cells doing the work.
Right.
But von Behring and Kitasato showed that serum, the liquid part of blood from rabbits immunized against tetanus or diphtheria, could actually prevent those diseases in other non-immunized animals.
Wow. So something in the liquid.
Exactly. Something tangible and soluble in the blood, not just cells, was responsible for protection. It was the first solid proof that these protective molecules, antibodies, physically existed.
So they proved something was there. Then, nearly 50 years later? 1939.
Yes, Arnie Tiselius and Elvin Kabat, they took it a step further. Using rigorous biochemical techniques, they pinpointed exactly where this antibody activity was within the serum proteins.
Right.
They found it was concentrated in what they called the gamma globulin fraction. So it wasn't just observing an effect anymore, it was biochemically isolating the molecules responsible, like going from knowing a car drives to actually identifying its engine.
Right, isolating the key component. And then Porter came along.
Yes, Rodney Porter in 1959, a really insightful contribution. He took IgG, which we'll get into, it's the most common antibody type, and used an enzyme, papain, to essentially cut the antibody molecule into distinct pieces.
Like molecular scissors.
Exactly. He identified 2 Fab fragments, that stands for fragment antigen binding, and 1 FC fragment crystallizable.
Fab and FFC.
This molecular dissection was absolutely critical. It showed how the antibody's elegant structure directly related to its different functions, like carefully dismantling a gadget to see how each part works.
It's amazing how these discoveries built on each other, piece by piece.
Brick by brick.
And finally, to sort of standardize everything, the World Health Organization stepped in.
That's right. In 1964, the WHO officially coined and standardized the term immunoglobulin. This was huge for global science, ensuring everyone was speaking the same language, which really helped foster clearer communication and faster progress.
So for you listening, what this really shows is that scientific breakthroughs are rarely that single eureka moment, are they?
Almost never.
It's more about relentless, step-by-step persistence. Each discovery isn't the final answer, but a key unlocking the next layer of complexity. It's how we got to the understanding and the medical tools we have today. It's really a testament to just keeping at it.
It absolutely highlights that collaborative, persistent nature of science. And speaking of pieces and structure, let's get into that iconic Y shape of the antibody.
Ah, yes, the classic diagram.
It's not just a nice shape for textbooks, it's a masterpiece of efficiency and function, perfectly designed for its job. That classic Y, that's the fundamental blueprint of a single antibody unit.
And what actually makes up that Y shape?
It's built from 4 protein chains. You have 2 identical longer chains called heavy or H chains.
Okay, heavy chain.
And 2 identical shorter chains called light or L chains.
Heavy and light. Got it.
And they're just floating near each other. They're intricately held together by strong chemical bonds, disulfide bonds like molecular rivets, plus a whole network of weaker but still crucial non-covalent attractions.
So it's a pretty sturdy structure.
Very sturdy. This robust construction is absolutely key to its stability and its ability to maintain that perfect Y shape ready for action.
Let's dig into those chains a bit more. The heavy chains are the longer ones.
Yes, quite substantial proteins, and their specific type is incredibly important because it's what actually determines the class of the antibody.
Ah, so the heavy chain defines the antibody type like IgG or IgM?
Precisely. IgM antibodies have what we call mu, heavy chains. Ig antibodies have gamma, heavy chains. IgA has alpha. IgD has delta. And IgE has epsilon, heavy chains.
Wow, okay. Greek letters for each.
Right. And these different heavy chain types are structurally unique, which allows each class of antibody to perform distinct, specialized roles in the body. They're built differently to do different jobs.
And what about the light chains, the shorter ones?
They're the partners connected to a heavy chain, completing the arms of that Y shape. Now here's a fascinating genetic detail. There are 2 types of light chains, kappa and lambda.
Kappa and lambda.
But, and this is the cool part, an individual antibody molecule will only ever have 2 kappa chains, or 2 lambda chains, never a mix.
Really? So it's either all kappa or all lambda on one antibody?
Exactly. You'll never find one of each on the same molecule. This genetic precision in how they're assembled is another layer of what makes each antibody so specific.
That ITER rule is pretty amazing.
Yeah.
Shows how precise the body's manufacturing process is.
It really does. And if we break down these chains even further, each chain, both heavy and light, has 2 distinct regions. There's an amino terminal variable, or V region.
Variable, meaning it changes.
Exactly. And then there's a carboxy terminal constant, or C region. This division of labor, variable versus constant, is fundamental to how antibodies manage both their incredible precision in recognizing targets and their diverse functions.
Okay, let's unpack that.
Yeah.
The variable regions are at the tips of the Y arms.
Right.
And they're highly variable.
Highly variable in their molecular sequence, yes. This variability isn't random, though. It's the ingenious mechanism that allows each antibody to create a unique binding site.
Within these variable regions, there are even more specific spots.
Yes, the hypervariable regions, sometimes called complementarity determining regions or CDRs.
CDRs.
Okay. These CDRs are truly the business end for recognizing the antigen. Think of them as the unique teeth of a key that have to fit perfectly into the grooves of the lock-in in the antigen.
So that's where the superspecificity comes from.
That's exactly where it comes from. Their specific molecular sequences enable that exquisite, precise binding. This is where the lock and key analogy really holds true, allowing each antibody to recognize its specific target with amazing accuracy, even telling apart molecules that look very similar.
Okay, so variable regions, recognition, and binding. What about the constant regions? They're more consistent.
As the name suggests, yes. These portions are relatively consistent within a given antibody class or subclass. They don't bind the antigen.
So what do?
They are absolutely critical because they determine the antibody's biological activities. Basically, after the antibody binds its antigen using the variable regions, the constant region dictates what happens next.
Like calling for backup.
Exactly. Does it call for backup? Does it flag the target for destruction? Does it trigger other immune responses? This is where the antibody's effector functions lie. It determines how it carries out its protective mission.
And this ties back to Porter's discovery, cutting the antibody into Fab and Fc.
Perfectly. His breakthrough really clarified this functional split. The Fab, the fragment antigen binding, is formed by those variable regions of both heavy and light chains. Its job is clear: latch onto the antigen.
The grabber part.
The grabber part. And crucially, each basic antibody unit has 2 Fab fragments. It's bivalent.
Meaning it can grab 2 things at once.
2 identical targets on an antigen simultaneously. This really enhances the overall binding strength, like having 2 strong grippers instead of just 1. Much harder to shake off.
Makes sense. So if Fab is the grabber, what about FEAC, the fragment crystallizable?
The Fc is made of the constant portions of the heavy chains forming the stem of the Y. And remember, the FTC region does not bind antigen.
Right.
Yeah.
No grab it.
Its real importance is being the command center for the antibody's biological activities. This is the business end for activating other parts of the immune system.
Like what?
For instance, the FVFC region can bind to that powerful system of proteins called complement, amplifying the immune response. It also facilitates something truly remarkable: placental transfer.
You mentioned that, allowing antibodies from mother to baby.
Exactly. IgG antibodies cross the placenta via their FVC region, protecting the unborn child. It's a profound evolutionary adaptation for early life immunity, and the FEC region interacts with specific docking stations, or FEAC receptors, on various immune cells, essentially telling them to take action.
So it's the communication hub telling other cells what to do.
Precisely. And connecting these pieces, the Fab arms and the FEC stem, is the hinge region.
The hinge, like on a door.
Sort of. It's a crucial, flexible area in the heavy chain's constant region, right where the arms diverge from the stem. Its purpose is vital: flexibility in binding.
Why is flexibility important?
Think of it like a universal joint. It lets the Fab arms move and pivot. This allows the antibody to act and bind optimally to antigens, even if they're on a curved cell surface or spaced out awkwardly. Without that flex, the lock and key might not always line up perfectly.
So it helps it bind better in tricky situations. It really is an elegant design.
A marvel of biological engineering.
But that Y shape is just the basic unit, right? Our immune system has a whole antibody army.
It does indeed, comprised of 5 distinct immunoglobulin classes. Think of them as different specialized units within a highly coordinated defense force.
Specialized units, I like that.
Each has its own unique structural variations, properties, and very specific missions tailored to different types of threats or locations in the body. Understanding these classes is absolutely key to grasping the diverse roles these molecular defenders play in keeping you healthy.
So what are the 5 classes?
They are immunoglobulin G, IgG, immunoglobulin A, IgA, immunoglobulin M, IgM, immunoglobulin D, IgD, and immunoglobulin E, IgE.
IgG, IgA, IgM, IgD, okay.
And the main differences between them come down to those heavy chains we talked about. The type of heavy chain dictates their structure, their properties, and most importantly, their major functions. Its form perfectly suiting function for each specific challenge.
Let's start with IgG then. You called it the long-lasting defender.
That's a good way to put it. Structurally, it's a monomer, a single Y-shaped unit, and it even has 4 slightly varied subclasses, IgG1 through 4.
And properties, you said it was common.
It's the undisputed heavyweight champion in terms of abundance, makes up about 80% of all immunoglobulins in your serum. It's the primary circulating antibody, constantly patrolling your bloodstream.
80%, that's a lot.
It is, and it lasts a long time too. IgG has the longest half-life, typically around 23 days. This sustained presence means it provides long-term protection.
And its main job?
It's the primary workhorse of the secondary immune response, the memory response. When you encounter something you've seen before, IgG dominates. It directly neutralizes toxins and viruses, blocking them from harming cells like a molecular shield.
And it does that tagging thing too, opsonization.
Yes, absolutely vital mechanism. IgG coats microorganisms, tagging them. This makes them much more easily recognized and then gobbled up by phagocytic cells like macrophages. Think of it as painting a bright target on the invader, speeding up the cleanup process.
So it helps the cleanup crew find the bad guys.
Precisely. And it also activates the classical complement pathway, that powerful cascade of immune proteins leading to pathogen destruction.
But it also has that unique superpower.
Oh, yes, the placental transfer. IgG is the only immunoglobulin class that can cross the placenta. This provides passive immunity from mother to fetus and newborn, crucial protection during those vulnerable first months, a truly remarkable adaptation.
Incredible. What about IgA, the mucosal guardian?
Right. IgA. In serum, it's a monomer, but its most important form is as a dimer, two Y units joined together called secretory IgA or SIgA. That's the form found in secretions. It also has two subclasses.
So it's mainly found where?
Its true importance isn't in the blood, but on the body's vast mucosal surfaces. Think saliva, breast milk, tears, the mucus lining your airways, gut, urinary tract. It provides localized defense right at the entry point.
The first line of defense.
Exactly, a crucial first line. And secretory IgA has a special component that protects it from being broken down by enzymes, which is vital for it to work in harsh places like the gut.
Built tough for the front lines. Does it activate complement like IgG?
No, it doesn't activate the classical pathway, though it can activate the alternative pathway. Its main jobs are agglutination, clumping bacteria together and blocking viruses from entering cells, essentially trapping and neutralizing threats right where they try to get in. The gatekeeper.
Okay, gatekeeper IgA. Next up, IgM, the first responder.
IgM, yes.
Yeah.
Its structure is quite something. In serum, it's a giant pentamer, 5 Y-shaped units joined in a circle.
5 units together. Wow.
Which gives it a theoretical valency of 10 binding sites, though it's often effectively 5 due to crowding. Importantly, it also exists as a monomer on the surface of B cells, acting as the B cell receptor, the initial antigen sensor.
So it senses trouble first.
It's key for that initial detection. Property-wise, it's about 5-8% of serum ego, with a short half-life, maybe 5 days. But its function is huge. IgM is the major antibody of the primary immune response.
The first time you encounter something new.
Exactly. When your body sees a new pathogen for the first time, IgM gets the ball rolling. It's the first wave of defense.
And its big structure helps.
Immensely. That pentameric shape makes it incredibly good at agglutination, clumping lots of pathogens together quickly. And it's a very potent activator of the classical complement pathway. Think of it as the swift, powerful opening attack against a novel threat.
Powerful first strike. Oh wait, what about IgD, the enigmatic sensor?
IgD. Structurally, it's a monomer. Properties. Found in really tiny amounts in serum, 0.2%. Short half-life 2 to 8 days.
So, what does it do?
Well, its primary known location is on the surface of mature B cells alongside IgM, acting as another antigen recognition receptor. But honestly, its precise, broader role in immunity is still somewhat elusive.
A bit of a mystery.
It is a captivating area of ongoing research. It reminds us there's still so much to learn about our own biology.
Okay, the mysterious IgD. And finally, IgE. The allergy and parasite fighter.
IgE, also a monomer. Properties are distinct. Extremely low amounts in serum, just 0.0002%.
Tiny amount.
Which highlights how potent its effects are. It's heat labile. And notably, it does not cross the placenta, activate complement, or typically agglutinate. Its roles are different.
Parasites and allergies, you said?
Its primary evolutionary role seems to be fighting helminthic parasites, worms. But for many people, its main significance is in allergies.
How does it work?
IgE binds tightly, via its Fc region, to receptors on mast cells and basophils. When this bound IgE then encounters its specific antigen, like pollen or peanut protein BAM, it triggers the rapid release of inflammatory mediators, like histamine.
Histamine. The stuff that causes sneezing and itching.
Exactly. That release is responsible for allergy symptoms and, in severe cases, systemic anaphylaxis type I hypersensitivity. So understanding IgE is vital for understanding allergies. It's a defense system kind of overreacting to harmless things.
Wow. So these classes are really specialized. But now, let's flip this. You said antibodies themselves can act as antigens. That sounds paradoxical.
It does, doesn't it? But yes. Because they're complex glycoproteins, immunoglobulins can be potent antigens themselves. They can trigger an immune response within the same body. It shows this incredible layer of internal regulation.
So our own defenders can become targets.
Under certain circumstances, yes. And because of this, immunoglobulins carry 3 major categories of antigenic determinants, like unique molecular fingerprints.
Okay, what are they?
First, isotypes. These are shared by all members of the same species. They're found in the constant regions and define the different classes and subclasses, like human IgG versus mouse IgG.
So isotypes define the class and species.
Got it. Second, allotypes. These are individual specific determinants found within a species. Think of them as subtle genetic variations between people's antibodies.
So my allotypes might be different from yours.
Potentially, yes. They're genetically controlled, and they actually have practical uses, like in paternity testing or population genetics.
Interesting. And the third type?
Idiotypes. These refer to the unique signatures found in the hypervariable regions, the Fab part, specifically at or near the antigen binding site.
So unique to that specific antibody's key.
Exactly. The fingerprint of that particular antibody's binding site. And the immune system can even make anti-idiotypic antibodies that recognize these idiotypes, hinting at complex regulatory networks controlling the controllers.
A system regulating itself. Amazing. Okay, we've got structure, classes, fingerprints. How are they actually made? The orchestra of production.
Right. The primary producers are B lymphocytes and their descendants, the plasma cells, but it's not a solo act. It usually needs the complex cooperation of 3 key cell types.
3 types.
Yes. You need macrophages, acting as antigen-presenting cells, APCs, the scouts. You need helper T cells, the orchestrators, the conductors, and the B cells themselves, the antibody factories.
Scouts, conductors, and factories. Okay, walk us through it.
It starts with antigen processing and presentation. An APC, like a macrophage, grabs an antigen, internalizes it, and processes it into smaller peptide fragments.
Chops it up.
Pretty much. Then it displays these fragments on its surface using special molecules called MHC class II molecules, like putting up a wanted poster for T cells to see.
So the T cells see the wanted poster. Then what?
Then comes T and B cell activation. Helper T cells recognize that antigen presented by the APC, but recognition alone isn't enough. It also needs co-stimulatory signals.
Like a password.
Kind of. A second signal, a handshake. It's a safety check to ensure the response is appropriate and prevent accidental activation against, say, our own tissues.
Makes sense. It's T cell activated.
Activated helper T cells then produce vital signaling molecules called cytokines. Think of them as messages sent out to stimulate the B cells.
And the G cells have already seen the antigen too.
Yes, B cells bind the antigen directly to their B cell receptors. Now, activated by cytokines in direct contact with helper T cells, they undergo rapid clonal proliferation.
Meaning they multiply like crazy.
Exactly, making a huge clone of identical cells, all specific for that one antigen. Then these activated B cells differentiate into 2 key types.
Okay.
Antibody-secreting plasma cells, the factories pumping out antibodies right now to fight the infection, and long-lived memory B cells, the library, storing the memory for future protection.
Plasma cells for now, memory cells for later. Clever.
Very. And there's another clever trick. Isotype switching.
Switching types.
Yes. Initially, B cells mostly make IgM, the first responder, but guided by those cytokines from helper T cells, they can switch to making other classes like IgG, IgA, or IgE.
But they still target the same antigen.
Exactly the same antigen. It's an incredible adaptive mechanism, tailoring the antibody type to the specific threat or location. Need long-term blood protection. Switch to IgG. Need mucosal defense. Switch to IgA.
That is amazing flexibility.
It really is. And this leads to the big difference between the primary versus secondary immune response.
First time versus repeat encounter.
Precisely. The primary response, the first time you see an antigen, has a longer lag phase, takes time to ramp up. Antibody levels are lower, mostly IgM. It's the learning phase.
Learning the ropes.
Then the secondary response on subsequent exposures to the same antigen. Much faster, much stronger, and predominantly high levels of IgG. That's immunological memory in action.
The basis of vaccines.
Absolutely. Why you rarely get the same cold twice, why vaccines give long-term protection, the immune system remembers and hits back harder and faster. I remember you, and this time, I'm ready.
It's incredible. And you mentioned things like age and nutrition can affect this whole process.
Yes, many factors can influence antibody production: genetics, age, nutrition, the dose and route of antigen exposure, even adjuvants used in vaccines to boost the response. It's a complex interplay.
Okay, so we know how they're built and made. Now let's get to the battleground. How do they actually protect us after they bind?
Right. Binding is step one. The action comes next. They have a diverse arsenal of mechanisms. This is the active phase.
What's the most direct way?
Neutralization. Antibodies bind directly to soluble toxins or viruses, physically blocking them from binding to our cells.
To stop them in their tracks.
Exactly. Neutralize their harmful effects before they can start, like putting a cap on a dangerous chemical bottle. Simple, potent.
Okay.
Opsonization. Tagging for destruction. Antibodies, especially IgG, coat pathogens. This coating acts like a flag, making them much easier for phagocytic cells, the cleanup crew, to recognize and ingest.
So they paint a target on the bad guy.
That's a great way to put it. Enhances phagocytosis, speeds up removal of threats, hugely important.
What about that complement system?
Complement activation. IgG and IgM are great at this. They trigger a complex cascade of serum proteins, the complement system.
And what does that cascade do?
Its ultimate power is lysis, punching holes in target cells like bacteria or infected cells, destroying them directly. But it also promotes inflammation and enhances phagocytosis further, like calling in an artillery strike that also signals ground troops.
Yeah, multi-pronged attack. Is there more?
Yes, one more main one. Antibody-dependent cell-mediated cytotoxicity, or ADCC.
ADCC.
Sounds complex. It's elegant, really. Involves teamwork. Certain killer cells, like natural killer NK cells, have FC receptors.
The docking stations.
Right. When an antibody binds to a target cell, say a virus-infected cell, its FC region sticks out. An NK cell can then dock onto that FC region.
And then?
That docking triggers the NK cell to kill the target cell. The antibody acts as a bridge, a targeting beacon, guiding the killer cell precisely to the cell that needs eliminating. Highly specific, highly efficient.
Like a guided missile system. Incredible versatility.
It really is. Neutralization, opsonization, complement activation, ADCC, a multi-layered defense.
So let's zoom in on that binding itself. The molecular dance. How specific is it, really?
The interaction is renowned for being highly specific, often compared to an enzyme and its substrate. But there's a key difference.
Which is?
Unlike enzymes, there's no irreversible chemical change to either the antigen or the antibody. They bind, interact, and can let go.
It's reversible. How does that work?
It's not one strong chemical bond. It's the sum of many weaker intermolecular forces, electrostatic bonds, hydrogen bonds, van der Waals forces, hydrophobic interactions, lots of tiny attractions adding up.
So it depends on a really good fit.
Exactly. The goodness of fit, the complementarity between the antigen's epitope and the antibody's paratope, determines the stability and strength. A perfect molecular embrace.
You mentioned strength. Are there different ways to measure it? Affinity and avidity.
Yes, two important terms, often confused. Affinity is the strength of attraction between a single binding site and a single epitope. One-on-one interaction strength.
Okay, affinity is one-on-one. What's avidity?
Avidity is the overall strength of binding when you have multiple binding sites on an antibody interacting with multiple epitopes on an antigen. Ah, like that IgM pentamer with its multiple arms.
Exactly. Even if each individual arm's affinity isn't super high, the fact that it has multiple points of contact gives it very high overall avidity. It's like trying to pull off something held by 10 hands versus just one much harder. That cumulative strength is avidity.
Makes sense. Now, you said they're highly specific, but sometimes they cross-react.
Yes. Specificity and cross-reactivity. While generally specific, antibodies can sometimes bind to apparently unrelated antigens.
Why would they do that?
Usually because of subtle structural similarities between the different antigens at the molecular level. Enough similarity for the antibody's key to partially fit the wrong lock.
And that can cause problems.
It can. In diagnostic tests, it might cause a false positive result. More seriously, it's thought to play a role in some autoimmune diseases, where the immune system mistakenly attacks self-tissues because they look similar to a foreign invader it was fighting. A real double-edged sword.
Delicate balance.
Yeah.
Now, you talked about stages of reaction, primary and secondary. Yes. The primary stage is the initial rapid reversible binding. Antigen meets antibody. Molecular recognition. But, crucially, there are no visible effects at this stage. It's all happening invisibly at the molecular level.
The quiet handshake.
Exactly. The secondary stage comes next. This is where subsequent interactions lead to visible effects, and these are what we rely on in the lab.
Like,
Things like agglutination, the visible clumping of cells or particles, precipitation where soluble antigens form visible insoluble complexes, neutralization becoming apparent, complement fixation being detectable, even seeing bacteria stop moving immobilization.
and these visible effects are the basis for medical tests.
Absolutely. They form the basis for countless serological tests. These tests harness antigen-antibody reactions to detect either antigens, is the pathogen present, or antibodies, have you been infected, exposed?
So they're fundamental for diagnosing.
Hugely fundamental. Diagnosing infectious diseases, autoimmune conditions, blood typing, tissue matching for transplants relies heavily on these principles. Think of tests like agglutination tests, precipitation tests, ELISA.
ELISA is a common one.
Very common, very versatile. And when you run these tests, you often measure the titer.
Titer.
It indicates the highest dilution of the patient's serum that still gives a positive reaction. It's a measure of how much antibody is present, the strength of the immune response.
Okay. But are there pitfalls? Complications?
Oh, yes. There are caveats and complications. One classic is the prozone phenomenon.
Prozone.
This is where having way too much antibody or way too much antigen can actually inhibit the visible agglutination or precipitation. Paradoxically, you get a false negative result because the ratio is wrong for forming those large, visible complexes.
Too much of a good thing can mess up the test.
Exactly. Need that Goldilocks zone ratio. And then there are blocking antibodies.
Blocking.
Some antibodies bind the antigen just fine, but don't cause agglutination themselves. Worse, they can get in the way and stop other antibodies, the ones that should cause clumping, from doing so. Another way to get a misleading result.
It's definitely intricate.
Okay.
Finally, what about when things go wrong? Abnormal immunoglobulins.
Right. Sometimes the production or the structure goes awry. Conditions like heavy chain disease, cryoglobulinemia. But a key example is multiple myeloma.
A type of cancer.
Yes, a cancer where a single clone of plasma cells grows uncontrollably. They churn out huge amounts of identical monoclonal antibodies, or M proteins, all specific for one thing, flooding the system.
That sounds bad.
It is for the patient, but historically, studying these uniform M proteins was incredibly important for science.
How so?
Because myeloma patients produced vast quantities of a single type of antibody, it gave scientists pure samples to study immunoglobulin structure and function back in the mid-20th century, when getting pure antibodies was extremely difficult. This natural experiment provided crucial insights.
So even from disease, knowledge was gained. A poignant point.
It is a reminder of how science progresses sometimes.
And that brings us full circle. We started asking about hidden defenders, and hopefully we've journeyed much deeper into the incredible world of antibodies. You can really appreciate their complexity, their precision, their vital importance. These tiny Y-shaped molecules are truly wonders of biological engineering.
Absolutely. And this understanding isn't just, interesting trivia. It directly informs so much of modern medicine. Vaccines, diagnostic tests, treatments for autoimmune diseases. It all relies on understanding these fundamentals of how your immunity works. It really fosters an appreciation for the body's defense mechanisms.
Definitely. So as we wrap up, here's a thought for you to ponder. Given this incredible specificity and adaptability of antibodies, and how much more we're learning about their rules, how might this continue to revolutionize personalized medicine? How might it boost our fight against new pathogens in ways we can't even imagine today?
The possibilities seem vast, don't they? We hope this deep dive has given you a newfound appreciation for these silent guardians within you. Keep exploring, keep questioning, and keep marveling at your own immune system.